Hello, how are you? Welcome to our discussion today. In this particular session, we shall talk about the process of seed germination. So in this particular discussion, we shall actually discuss about the conditions that the definition of seed germinations, and also we shall talk about the conditions that are necessary for the process of germination to take place. And finally, we shall talk about the changes that occur inside the seed that will bring about seed germination. So, quick question here is, what do you understand by the term seed germination? What is seed germination? Okay, the process whereby seeds develop into a seedling is what is called seed germination. So seed germination refers to the process by which seed develops into a seedling. Process by which seed develops into a seedling is what is called seed germination. Now, what do you understand by the term viability? So viability of a seed is the ability of a seed to germinate. Some seeds cannot germinate because they're not viable. So seed viability refers to the process by which, okay, the ability of the seed to germinate. Okay, good. Now, what do you think are some of the conditions necessary for the seeds to germinate? What are some of the conditions necessary for a seed to germinate? Yes. Can you name some? So you might have mentioned some conditions like water, oxygen, warmth or temperature, enzymes, hormones, and viability. So all these are the conditions that are necessary for germination to take place. So we shall discuss the function or the role of all these conditions, which are very important in seed germination. Now, let us first of all talk about water. What do you think water has to do with germination? Yes? So first, water is a medium of enzymatic activities. This means that the enzymes perform their function well where water is available or present. Yeah, because it is a medium for enzymatic activity. Number two, it brings about hydrolysis of food into simpler substances. You know, the food stored inside the cotyledons are actually in form of the complex food substances like the polypeptides for the proteins or polysaccharides for the carbohydrates. So for this food to be broken down into simpler forms, we learned in Form 1 that the process known as hydrolysis will occur. So hydrolysis will require water for the food substances to be broken down. That's why we are saying that for seed germination to take place, water must be present. So water brings about hydrolysis of food into simpler substances. Also, water is a medium of transport. After hydrolysis has occurred, the end products of hydrolysis have to be taken to the growing regions. That is the root tip and the shoot tip, where the cells are dividing actively. So what will take these end products of hydrolysis? Water. So water is actually a medium of transport of the dissolved materials or hydrolyzed materials to the growing zones. It also softens the seed. For seed to burst open, releasing the radicle, the seed coat has to be softened. And it is softened by by water. Then it also acts as a solvent. Water acts as a solvent of many substances which are required during germination. Above all, number 2, the other factor is oxygen. What do you think oxygen is needed for during germination? Now, oxygen is needed for the oxidation of the food substances or respiration to take place. You know, for growth or germination to take place, the seeds require energy. Where will the seedlings get energy from? You will say from the food that is stored in the cotyledons, yes, but the food has to be broken down into simpler forms. The food has to be converted into energy through respiration. So the food therefore has to be oxidized by oxygen. So oxygen is needed, 1, in order to oxidize the food so as to release energy. In order to oxidize the food so as to release energy. This energy is needed for cell division and growth. Okay. Apart from that, we also need suitable or optimum temperature. And this optimum temperature is about 30 degrees Celsius. What do you think is the role of the optimum temperature? Good. Earlier, we have talked about the processes that occur here during germination, respiration. Respiration is enzyme controlled. This means that it is controlled by enzymes. And enzymes work best at optimum temperature. We learned in Form 1 that when temperature goes beyond the optimum temperature or above the optimum temperature, what will happen to the enzymes? Enzymes will be denatured. What if the temperature is below the optimum? It will inactivate the enzymes. Therefore, the seeds, for them to germinate, the temperature must be maintained at around 30 degrees Celsius. That is the optimum temperature. So we've said that the optimum temperature will activate enzymes involved in mobilization of food reserves. Apart from that, we have also learned that lower temperature activate enzyme, lower temperature will tend to inactivate enzyme, while high temperature will denature the enzymes. And apart from that, it will also kill the protoplasm. Protoplasm refers to the cytoplasm and the cell sap and all the organelles that are found within the cell. That is what is actually collectively known as protoplasm. So when you have higher temperature will denature the enzymes. And apart from that, it will also kill the protoplasm. On the other hand, lower temperature will inactivate the enzyme. It will make the enzymes needed for germination to take place to become inactive. Good. Then another factor is viability. Viability. We have said that viability is the ability of the seed to germinate. The ability of the seed to germinate. So this means that only viable seeds are able to germinate and grow. Only those viable seeds are able to germinate and grow. Seeds stored for long period usually lose their viability due to depletion of their food reserves and destruction of their embryos by some pests and diseases. So we are saying that for the seeds to germinate, they must be viable. And we have seen here that when the seeds are stored for a long period of time, they will lose their viability. They no longer germinate when grown, okay? Because they'll have lost their viability, okay? So if they lose their viability or the food reserves might have been depleted, this will make the seeds not to germinate, and they might be destroyed by the pests and diseases, hence losing their viability. So in a nutshell, we have said that seeds, though for those seeds which are viable, they are the ones which will germinate. Okay. Now, another factor which we ought to discuss is enzymes. For germination to take place, there must be some enzymes. These enzymes are very important in 1, mobilization of the food. the food which has been stored in the cytoplasm has to be hydrolyzed. Has to be hydrolyzed. So for hydrolysis to take place, enzymes are required. Apart from that, we also have assimilation of the food. That is, the food has to be broken down to release energy through the process of respiration. So for germination to take place, enzymes must be present. We have different categories of enzymes, as you shall see here. So we are saying that enzymes play an important role in the breakdown and subsequent oxidation of food. Stored food is in a form of carbohydrates, fats, and proteins, which all of them are insoluble. The carbohydrates have to be broken down to glucose by enzyme diastase. So during germination, enzyme diastase is produced. This enzyme will break down carbohydrates into glucose, which is mobilized or taken to the growing regions, that is the plumule and the radicle, the root tip and the shoot tip. Okay. Fats are broken down to fatty acids and glycerol, the building blocks of fats, or the building blocks of fats, and also by lipases. The fat are broken down by enzymes called lipase, while proteins are broken down to amino acids by proteases. So these end products, that is glucose, fatty acids and glycerol, and the proteins and amino acids, from proteins are usually taken to the growing regions, whereby some of them, like the carbohydrates, the glucose, will be broken down into to release energy. And some of this will be used in the synthesis of new cells, especially the cell membrane and cell walls. Also, the fatty acids and glycerol and amino acids will be used in the synthesis of new organelles. So we are saying that the enzymes that break down carbohydrates to glucose is called diastase. That which breaks down fats to fatty acids and glycerol is known as a lipase. And those which break down proteins to amino acids are proteases or protease. Okay. So apart from breakdown of the stored food in cotyledons, enzymes also facilitate conversion of the hydrolyzed food products into new plant tissues. Okay, like new cell walls, new cell membrane, new organelles. This will be facilitated by those enzymes. Good. So far so good. We have learned about the factors that determine or that is necessary for germination to take place. So can you still remember? So we've talked about factors like enzymes. We've also talked about factors like viability, temperature, oxygen, and water. Good. So I think we shall stop there, because in the next session now, we shall discuss about germination, the process of germination. That is, what are some of the physiological changes that occur during germination. See you again. Bye-bye. Have a nice time. Welcome back. Now, let us discuss about the physiological changes that occur in a seed during germination. For us to discuss that particular aspect, let us first of all have a look at this short video.
What is...
Sorry. Okay.
What is germination? When a seed is sown into moist soil, it begins to grow. This process is called germination. It occurs in the following steps. The seed coat splits. A tiny root grows downward and a shoot grows upward. This produces a seedling of a plant, which ultimately grows into a new plant. After some time, the plant bears flowers. The flowers produce fruits, and the fruits have seeds in them. All seeds can germinate when sown in moist soil under suitable conditions to produce new plants. By natural or man-made processes, these seeds are sown into the soil again, and that is how new plants are born.
Thank you. Thank you very much. Now, we have seen some changes that develop when actually germination occurs. But before we discuss those changes, please keep them in mind. Let us also have a look at this video. Good. Thank you very much for being actually keen and observing this particular video. Now, what happens when a seed is sown in the ground? We have seen that it is capable of, if it is viable, it will germinate into a new seedling. But what are some of the physiological changes that occur within the seed? So if you look in this particular glass, usually the seeds, upon actually being actually planted in the ground, and if the soil is moist, it will absorb some water vapor, not water vapor, but some water through the micropyle. Okay, through the micropyle. Then it swells up, a process called imbibition. We shall see later, imbibition. Then as it absorbs water, you know, the water, as we have learned earlier, it is needed for the process for the hydrolysis of food, which is stored inside the cotyledons. So as the end product of hydrolysis reaches the growing regions, it makes now the seedlings to start growing, where the cells will start to multiply at the radicle and the plumule. Then we have seen that the radicle will be the first one to emerge through the micropyle, and it moves downwards. Okay. Then as it goes downwards, we have seen that in this particular video, we have seen that there is this part here, the part between the radicle and the cotyledon. We have this part here. This is what we call the hypocotyle. The hypocotyle will tend to push upward. Okay, the hypocotyl will tend to push upward, hence making the seedling to emerge out of the soil by making the hypocotyl to extend. So we're saying that the radicle will grow downwards while the plumule will grow towards or upward. Is it clear? So you realize that we have two main types of germination. We shall see later. Okay. So what happens when the seed rises above the ground? As you can see here, it will come out. Some seeds, depending on which type of germination, will come out with the seed coat or the cotyledons. Okay, and then new seeds will start to form, or new leaves will start to form. Okay, some of the cotyledons or the cotyledons may also form green coloring matter called photosynthesis. Hence, the food will be able to photosynthesize. So that is the process, or that is what actually happens during germination. Let us now go to the literature. Good. So the physiological changes that occur in the seed during germination include: one, we have said that at the beginning of germination, water is absorbed into the seed through the micropyle, and this process is called imbibition. This causes the seed to swell. Then next is that in the presence of oxygen, optimum temperature, and water, food reserves in the seed are hydrolyzed or broken down into soluble diffusible form by enzymes, as we have learned earlier. Okay. Then the soluble food diffuse to the growing regions, that is the plumule and the radicle. Okay. Then oils and carbohydrates will tend to provide energy needed for the germination process, and simple sugars will be converted into cellulose to form new cell walls. Amino acids will make the protoplasm. Then the plumule grows into shoot upward, while radicle will grow into root downwards. Then the radicle will emerge from the seed through the micropyle, hence bursting the seed coat. And that is what happens during germination. So first is that the seed will absorb water through micropyle, a process called imbibition, and this will cause the seed to swell. Then if there is presence of oxygen, optimum temperature, and presence of water, the food reserves in the cotyledons will be hydrolyzed, will be broken down into their simpler forms. Okay, then they'll be taken. The soluble food will diffuse to the growing regions of the embryo, and then they will be assimilated. In that, oils and carbohydrates will provide energy. Simple sugars will be converted into cellulose to form cell wall, and amino acids will make the protoplasm. Then in the process, as growth occurs, cell division occurs, the radicle will come out to form now the roots or the root, while the plumule will come up, will come out to form the new shoot. The radicle will emerge first, and it will emerge through the micropyle, hence bursting the seed coat. Okay, so bye-bye, have a nice time. Let us meet again in your next session so that we can talk about the types of germination. Have a nice day or nice time. See you again. Bye.